Welcome to Notes from the Lab, an ongoing series of conversations on the past, present, and future of the BCG vaccine. My name is Denise Faustman, and I'm head of the immunobiology labs here at Mass General Hospital and Harvard Med School in Boston. In this podcast series, we're exploring topics related to type 1 diabetes and the BCG vaccine and how this all fits together, of course, with tuberculosis as well. My guest today is from McGill University in Montreal, and he's a professor of medicine and, among other things, also the associate director of the McGill International Tuberculosis Center. So, Maz, why don't you introduce yourself and we'll go from there.
Yeah, sure. My name is Maziar Divangahi. I'm a professor of medicine at McGill University, and for the last 25 years, I'm working on figuring out a way to develop kind of a novel vaccine against tuberculosis. But of course, as Dennis mentioned, one of the common vaccine that we have, I mean, the only vaccine actually we have for TB is the BCG. And also BCG does many other things that we still don't understand. So subsequently, I think understanding how BCG works gonna help us to find not only a new vaccine for TB, but also how we could harness the power of BCG against other infectious diseases or against other autoimmune diseases. And I think that's really important to understand.
Mhm. And just if you're part of the lay audience here, the BCG vaccine is like tuberculosis of cows. And so they're very similar, other than TB or tuberculosis is for humans, is most infectious in humans. And Mycobacterium bovis, which is the BCG vaccine, was most infectious in cows and then subsequently 100 years ago became the BCG vaccine. So that's the connection we're going to talk about today. So,
Yeah,
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 go
go ahead.
ahead.
No, exactly. I think, the BCG, as you indicated, is attenuated form of the M. bovis, which cause TB-like disease in cows. And this vaccine has been discovered more than 100 years ago, and is effective in preventing disseminated form of the tuberculosis in children, but it will lose its efficacy in adult. And we still don't know what are the mechanism of protection against children. So there are still a lot of unknown about BCG, and I think as I indicated, I think understanding the cellular and molecular mechanism that involve in BCG could really help us to map out the hopefully the protection in against TB also. So if I want to start with the BCG, it's as I mentioned, it's been the only vaccine that we have is actually one of the safest vaccine that we have in the world because uh, neonate as soon as they born, at the age of 2, 3 days old, they get this vaccine, and the side effect of the vaccine is very, very little. I mean, it's almost close to nothing. So it's extremely safe. And if you go back to the old literature, we could see that BCG not only provide protection against TB in children, but also provide protection against other pulmonary infections, in particular viral infections. So again, these are all indicate that the impact of BCG on immune system is far beyond just providing protection against TB in children.
Hmm. But still, it's pretty good against children. That's the thing. It's something so old. Do you also, for this audience, want to talk a little bit about in the US and Canada, there's not much tuberculosis. And
Yeah.
so most people in the US have little appreciation of why that disease in and of itself is important. I think your background's in lung pulmonary medicine, right?
Yeah.
So, do you want to talk a little bit about TB?
Yeah. So, I mean, I'm a pulmonary immunologist, and as I said, I work on the pulmonary infectious diseases for quite a long time, in particular TB. And as you mentioned, TB is the disease of poor countries.
Yeah.
So it's not a disease of rich countries like US, Canada, and even Europe.
Hmm.
But if you think about a statistic and numbers, TB still kills around 1.5 million people every year.
Hmm.
So this is quite of a high number of people die of tuberculosis around the world, but maybe not in US and Canada, but in many other countries, highly endemic countries such as India, in Africa we have, in South Africa, in China to some extent. So, you know, we have moral obligation actually to really figure out how to prevent this disease. And unfortunately, the funding, because it's not a disease of the rich people and rich countries, the funding is very limited for this disease. Uh, and we need more funding because we still don't understand the pathogenesis of the disease, and thus we can come up with a novel vaccine against TB. The approach that we have basically is a conventional approach for developing vaccine. And we like what we do usually for viral infection, we overexpress one of the dominant antigen of the pathogen, and using viral vectors, or in this case of the COVID, we use messenger RNA, an RNA technique, vaccine-based technique. And we basically overexpress one antigen, we develop antigen-specific T and B cells, and and if we are lucky and we are not dealing with the complex diseases, then we are able to have a effective vaccine. And we got lucky with the SARS-CoV-2 because we
Hmm.
think it's not a very complex disease, and overexpressing of one antigen was enough to develop that antigen-specific B cell responses and neutralizing antibody against the virus. But this approach has not been successful in TB, uh, for multiple reasons. Because first of all, we don't understand what are these T cells are doing. So virtually all the vaccine that we are using, we are developing against TB, is targeting antigen-specific T cell responses, in particular CD4 T cells. The problem is that none of the preclinical and clinical studies using this approach has been successful, because we don't know what are these T cells are doing actually. So,
Hmm.
if there are literature, very strong literature that, if you're looking at the antigen-specific MTB, human antigen-specific T cell epitopes for MTB, they're evolutionary hyperconserved. So, if you think scientifically, any pathogen that has an antigen that is really could be targeted by the immune system and be bad for the pathogen, they will make a mutation. So the
Hmm.
mutation rate in MTB is extremely low. And in fact, some of these antigen-specific T cells have been shown that may help the disease and spread of the disease, and basically promoting the pathogenesis of the disease. So this type of a shotgun approach that we have used for MTB in terms of developing vaccines has not been successful. So we need to figure out, what are the correlates of protection against TB first, and then by identifying that, then we could, thinking about how to develop a vaccine. So I could give you an example. For instance, we know that BCG provide protection against children,
Hmm. Hmm.
but we still don't know what are the mechanisms. A lot of the studies thought that is antigen-specific T cells, conventional T cells, and all those studies suggest otherwise. So one of the things that we are actually decided to look in our program here at McGill is that how BCG provide protection in neonate and children, because the idea was if we could identify and map out this protective mechanism in children, then potentially we could harness the power of this in for developing a vaccine. So this study that one of my PhD students here is conducting is quite become interesting because what we found is that we established a mouse model because there is not any other way that you could study that in human. Is very complex to really sort out the cellular and molecular mechanism involving this protection. So so we established a mouse model as soon as a neonate was born, like in human, day 2, 3, we vaccinated with BCG. We have the control adult group also vaccinated with BCG. And we could see that in the mouse model, the adult, when they get vaccinated, they're able to have protection up to 3 months, 4 months, and after that, that protection is gone. But in neonate, when we vaccinate, now we have this protection going on all the way to 1 year. Okay. So, and you have to realize a mouse live up to 3 years, so that 1 year is really translate to quite a lot. So we were able to some extent reproduce what we see in human in a mouse model. And it become more interesting because we noticed that, when the route of the vaccination in human is intradermal. So in the mouse model, we also try to reproduce that we were giving it subcutaneously. And what we observe is that when we does subcutaneous vaccination in the mouse, we see that the bacteria actually disseminate through the blood into the multiple organs. Okay. And this was quite interesting and surprising to us because we also know that in adult, when you vaccinate intravascularly, you provide a better protection against MTB. And this is not only in the mouse model, because people in 70 also shown in the non-human primate, when you do intravascular BCG vaccination, that's a superior protection.
Yeah,
and
absolutely.
against any route of the vaccination. So we think that the dissemination of BCG is actually very important. And in fact, we start looking at the skin of the neonate versus adult, or even in human, it's been well established that the skin is very different in neonate than adult because the epidermis, dermis are much thinner and they are more vascularized. So you could imagine that if you put a needle into the skin of the neonate, it will hit a lot of those veins underneath. And that provide basically the route of the dissemination of BCG.
Speed.
Exactly. And if you go back to the literature, old literature in human, you could see that, after BCG vaccination in children, they found it in circulation, and it's kind of documented too. So just changing, just this route of vaccination or changing the route of vaccination could make a tremendous impact with the current available BCG vaccine.
Do you think you could get approval to go IV? That's the
Yeah.
$64 million question.
Yeah, I mean, it's really interesting, your question.
Yeah.
I mean, Bob Seeder at the NIH has done quite a lot of work in non-human primate. And beautifully show in the paper in Nature several years ago
Yeah, in
that if you,
primates. Yeah.
in primates, if you
Yeah.
do BCG IV vaccination, you could actually, in some primates, completely prevent infection. That means that infection didn't take a place from the, upper airway into the lung. So this is really amazing, because clearly, even in non-human primate, you see a better protection, independent of, the mechanism of protection. That could be T cells or innate
Hmm.
cells or a combination of both. We think that this makes a huge difference. Can we translate that to human? I think that's something that Bob is looking for to
Yeah.
try to see if he could conduct a small scale experiment. But I I think it's feasible because for two reasons. First of all, if we think that a neonate get BCG and it gets systemically and
Mhm.
in human, and that's why they are better protected against a TB. And if 2, 3 days old baby is able to, handle, 10 to the 6 CFU in the circulation, then um, that provide more rationale that this could be done in humans.
Mhm.
The other things is that MTB is a very different type of bacteria. MTB is a slow-growing bacteria.
Mhm.
MTB replicate every 24 hours. It's not like Pseudomonas or gram-negative bacteria that replicate every 20 minutes and
Yeah.
they
Yeah.
could cause
You can
sepsis
grow
and
out the next day and say you've got tuberculosis.
exactly and they cause sepsis and all of those things.
Yeah.
Uh, The other rationale that we have also is that, as you know, BCG is the only treatment that we have for bladder cancer.
Hmm. Hmm.
So, and in the bladder cancer, we are not talking about 1 million. So we're giving 10 to the 8, 10 to
Yeah.
the
Yeah.
9,
Massive. Massive.
massive dose, and not only one time. There is a whole protocol that every week you will get one up to 6 weeks, and then there's a maintenance dose. So these people get quite a lot of BCG. And we also know that BCG the way that BCG provide protection in bladder cancer is because it's able to disseminate
Oh,
again
yeah.
through the blood. One of the mechanisms that we describe is able to actually getting into the bone marrow, reprogram hematopoietic stem
Mhm.
cells, and then generate a very good kind of anti-tumor immunity, mainly dependent actually on innate cells, in particular neutrophils, macrophages, or DC, that are also able to provide better present antigen to the T cells. So clearly, these people are really, even they have cancer, they're immune compromised, and all of those things,
Mhm.
they're able to maintain the level of this number of the BCG systemically. So,
And safely. I mean, some
and
of them, anybody can get the vaccine and it goes systemic, which is really rare. And
yeah,
once in a while in the bladder clinic, right, they have a case where it goes,
BCGosis, yeah,
yeah, but it's rare.
very rare. I
And you
agree.
probably know these two, there's multiple papers on it of you give high dose, this is in diabetes, one of the off-target things we work on, as well as in dementia now. There's two big bladder. Actually, for Alzheimer's, there's 4 papers now showing if you have bladder cancer and you get these high doses of BCG, you don't develop dementia. The curves depart. And that's
Yeah,
from Israel as well as from MGH. And then in diabetes, we did the same thing. And you get immediate, in a year protection. Unlike when we're doing our trials, you got to wait 5 years for the full course. So I
yeah,
think there's already a lot of human data that when you're putting BCG, at least in adults, it's delayed. And in our two COVID trials done here in the US, I mean, nobody had BCG, right? But
Yeah.
we're really lucky these people had already had 6 doses of BCG a couple years before. When we do those timelines, and we keep giving those timelines to Christine, Ben, it takes 5, 6 years for infectious disease protection. So it
Yeah.
can occur in adults, but it's so slow with the intradermal wound.
Yeah. Yeah.
So I
No,
think all the data fits beautifully together. Yeah.
I totally agree. And as you indicate, there is more into BCG. I think, beside the protection in neonates for the
Hmm.
TB, like BCG provide protection against influenza virus.
Hmm.
And, we model this and we know that how actually it works. And, so there is a lot of dialogue between the innate compartment and adaptive compartment.
Hmm.
And this idea that memory is only limited to the adaptive arm of immunity, I think is really incorrect,
Hmm.
because we clearly see that you could induce memory in the innate compartment, and we
Hmm.
haven't really tapped on this idea for developing vaccine or for treatments and therapeutic approaches. There are tons of things we could do. And you could imagine that, if we could develop a vaccine that, generate a very strong innate memory response in the alveolar macrophages,
Hmm.
which are these are the cells that sitting in the lower part of the airway, and they get infected by MTB. And basically, MTB hijack their antimicrobial properties and grow within those cells, because MTB is intercellular pathogen.
Hmm.
So if we could reprogram somehow those alveolar macrophages that from get to go an earlier stage of infection before the bacteria has the chance to move into the lung tissue and form granuloma to,
Hmm.
basically
Hmm.
to wall off the infection. If we could really trap the bacteria in the airways with those cells and kill them, then the whole things would fall apart.
Yeah. Yeah. It
The,
would
the, the
stop early.
next, yeah, the next sequence will stop. We are never going to get into the point of getting to the granuloma, because that part is very complex. You have a mixed bag of the innate and adaptive immune cells that coming, and the bacteria is in the center, is trapped. How are we going to deal with it's very complex. So if we could deal with this infection at the very earliest stage of infection, then we could probably have a better chance to find a vaccine against TB.
Hmm. Do you think, and I don't know the literature because you're the TB expert here, I focus on BCG. So for BCG, we know in adults, it takes a long time, whether it's diabetes, whether it's glycolysis, whether it's dementia markers, whether it's the induction of Tregs, okay? And it's all in the lymphoid cells because we've done functional PET scans to show the spleen like lights up with glycolysis, but it takes 2 to 3 years. Do
Yeah.
you think in humans, the human trials with BCG in adults might not have, with intradermal, maybe didn't go long enough in time? I mean, we're going 5, 6, 8
Yeah.
years.
Yeah, I mean that's exactly what I think you're saying is correct,
Yeah,
because when you do intradermal vaccination in adult
yeah,
skin, the bacteria, the vaccine basically, or BCG, will end up in the lymphatic system.
yeah,
Yeah.
yeah.
So it wouldn't have a chance to getting systemically into the circulation and hit
Mhm.
other organs. So it will go through a very different route,
And
and
slower. And
much,
slower.
much slower,
Yeah,
absolutely.
yeah,
And then again, as we discussed, and BCG replicate every 24 hours.
yeah.
So you could imagine that how long it's going to take and where it is, and and
Hmm.
and so um if we could accelerate this process, like we have shown by, giving it systemically into the circulation, or in the neonate,
Hmm.
when they get access into the circulation, then everything will be different.
I think so. I
And then
think
you
so.
have and you have this like really amazing protection against multiple different things, you know.
Mhm.
And you know, every time we're looking at the effect of BCG that has been successful is somehow linked to its dissemination systemically. You
Mhm.
could see it with the BCG in the bladder. You could see it with the BCG vaccination against flu. You know,
Mhm.
all of those things that, you know, against TB, it really linked to the one point that can BCG get access into the circulation and disseminate to the multiple different organs,
Yeah.
including bone marrow, spleen, and other tissues.
I
So
think that was really the breaking point. I mean, the first time we realized the time course in adults was from Ristori's data in Italy when he was doing multiple sclerosis.
yeah,
We're doing 1 year trials, we're doing 22-week trials, and he's going 5 years in multiple sclerosis. And
yeah,
he sees these amazing CNS effects and lack of relapses. And it was like scratching our head and going, "You're going 5 years? Okay, this makes it easy. Okay, this makes it
yeah,
really easy to do trials." But that was kind of the breaking point for these other off-target things in unvaccinated adult population. You had to be so, so patient.
exactly,
Yeah.
exactly.
You
Yeah,
can't run out in a COVID pandemic, give somebody an intradermal COVID vaccine and hope you're going to see an effect.
yeah.
It's just
No,
not going to happen.
exactly. And I think, you know, as many vaccinologists know, the route of vaccination make a huge difference. I mean, you could see that, you know, the route of vaccination of intradermal versus IV with the
Mhm.
same vaccine suddenly make a tremendous difference
Difference. Yeah.
from preventing MTB and a disease to, you know, completely um, killing the pathogen.
Hmm.
And so it makes huge difference.
And durability of the
And the
response.
durability.
Yeah.
Yeah,
Yeah.
yeah,
Yeah.
yeah, yeah,
I remember hearing that a lot of the poor countries in Africa would take the American made vaccines. You pick your favorite, and they dilute them 1 to 10 or 1 to 20. And instead of going IM, they'd go intradermal. They go, well, that's much better. They
yeah,
get a better bang for their buck. But I think the next step from intradermal is probably IV if any human studies committee will
yeah,
allow that. Yeah.
exactly. And you know, to my knowledge, BCG is the only vaccine that is given intradermally. In
Hmm.
fact, initially BCG was given orally. So there
Hmm.
is lots of study that, you know, you will get into the GI tract with a
Hmm.
very high dose. And that was very effective also, again, because mainly probably disseminated to multiple organ.
Hmm.
But yeah, I mean, this is the only vaccine that is given intradermally.
Yeah. But I think people have tried a mouse IM and it doesn't work very well. But
Yeah,
yeah,
yeah.
again, it's about dissemination and
Exactly. And
it's
you know, again, so every route of the vaccination will trigger a different type of the immune response.
yeah.
So when we giving subcutaneously or intradermally, you
Hmm.
know, in an adult mice, not neonate,
Hmm.
they usually go into the lymphatic system, and
Hmm.
you know, they usually deal with the generating antigen-specific T cell responses. So
Hmm.
it's a very different route than if you're giving it IV, which
Yeah.
you know it goes to
Everything. It goes to
exactly,
lymph nodes, it goes to
exactly,
bone
yeah,
marrow, it goes to the organ.
yeah,
Absolutely. Well, we kind of, from our viewpoint, abuse the TB literature because every time we find something off target, we always go back to the tuberculosis literature and say, did somebody know this already? And certainly that's been the case so many times. Like we see all these glycolysis effects in human
yeah,
trials. And we go back, and there, lo and behold, in the granuloma is the Warburg effect happening
yeah,
every day.
yeah, yeah,
And then we see in autoimmune diseases these beautiful induction of Tregs, obviously a massive T cell response. And we go back to the tuberculosis literature, and you guys already said
yeah,
it's a pocket. The granuloma is a pocket of Tregs. So I think, not being part of the TB literature, I think TB is so tough because it's almost like cancer. You've got massive Treg response. So how do you get rid of those Tregs?
Yeah. And this is a very good point also, Dennis, that you're bringing up because those Treg is true that they will suppress the immune responses and they
Yeah,
try to
to kill it.
but
Yeah.
exactly. So, but at the same time, we need to do that
Hmm.
because otherwise, if that granuloma become totally inflamed,
Hmm.
that could kill the
The
host
host as well. Yeah.
because of lack of disease tolerance, we call it. So you could also argue that, human it's pretty good to deal with TB because, only 10% of the population develop active disease.
Hmm.
90%, which will, get to the point of
Or a
forming
carrier,
granuloma.
beautiful
And,
carrier.
exactly. So we kind of like find a way to live. And, a lot of these people eventually they die of cardiovascular diseases,
Mhm.
cancer, other diseases, but
Normal
not
things.
of TB if they're not become immune compromised. And, so I think those Tregs, they need to be there because they also need to control the immune system.
Hmm.
Otherwise, this dysregulated
The
immune system,
necrosis and stuff would be
exactly,
terrible.
this dysregulated immune system could be a major problem, and that would kill the host. I mean, we have established model system, and other people that if you really dysregulate, let's say, antigen-specific T cell responses against MTB, which we think is a major component of killing MTB, you actually compromise the host survival because you increase this immunopathology in the vital
Mhm.
organs such as lung. So a lot of like, I mean, this PD-1 blockade, and things
Yeah.
that will completely unleash T cell responses, it turned out that, it will activate TB, like cancer
Interesting,
patient that they
interesting.
get,
So on surface, it looks so good, and then you do it, and you go, maybe not so good.
exactly, exactly. So I mean, there is a quite of a, very good balance between immune system and MTB, and changing that balance could also become problematic. So if you're looking for the vaccine and you want to get rid of this pathogen, I think we must think about the timing that the bacteria still is in the airway
Hmm.
and hasn't really moved into the lung tissue. So that is the time frame that we have. And I really don't know what is that time frame in human. That's not been like clear to me. But that's the best time. That's
To
the best
intervene,
window
yeah.
we have to really eliminate the pathogen.
But on the other hand, maybe TV is good for us. If BCG is so good for us, I mean, I'm sure you saw this article. There was an
Yeah.
article that all these geneticists went after. It was from France, and I think it was in Nature too. And they were spearlunking to get down these caves. And these guys, this is about 6, 7 years ago, looked over and they go, "There's Joe. Oh my God, he fell down this cave a long time ago. He must be 100 years old." So of course, everybody went to get this guy, this freeze dried, down deep in the earth guy out.
Yeah.
Turned out he was a Neanderthal. Okay,
Oh my God. Yeah.
he hadn't fallen down 100 years ago. He had fallen down 100,000 years ago.
Wow.
So there was this huge rush to get his DNA out. Can you really finally get whole sequence DNA? And you know what they got? They got whole sequence tuberculosis out.
Yeah. Yeah. I'm not
And
surprised.
and so TB has evolved with us to the Neanderthal stage, if not before.
Yeah.
So I think some of these health benefits we're now seeing with BCG are why TB is synergistic most days, but not all days. It's a good relationship, but it's not perfect. And
Yeah. No, I think this could be true. I mean, but the problem is that, you know, 90 to 95% we are good to control MTB, and they might have some benefit that we don't know. And there are
Mhm.
also studies suggesting that, they increase other things like, COPD and
Yeah,
other issues. But the problem is that 10%, 5 to 10%, because they develop active disease and because the sheer number of those 5, 10% is so high,
yeah.
then it still translates to 1.5 million
Deaths,
deaths
deaths.
every
It's
year.
kind of like sickle cell anemia. If you're in
Yeah,
Africa, it's a good thing to have. If you're in Canada and the US, not so good.
no, no,
Or
exactly.
thalassemia in the Mediterranean and
Yeah.
malaria. So there's some of these things. So I'm just
But but
hoping,
but you're right. I mean, we evolved with TB for so
yeah,
many years, and clearly, there is
there's benefit.
yeah, there's a
There's
long,
benefit.
kind of interaction between the bacteria and the
Yeah,
host and So yeah, I mean, there are tons we still don't know about this bacteria. Yeah.
yeah,
You could argue that BCG might be a very beneficial in multiple different reasons, and because historically, as we discussed, is the safest vaccine is given, to the children and this and that. Um, and you know, we don't use BCG in our children anymore.
no.
In that there are, and I don't understand why. Like, I mean, I really wanted to vaccinate my children with BCG,
Yeah, it was the one
and,
vaccine you
and
really wanted, but it's
yeah,
not available.
yeah, exactly, and I couldn't. So
Mhm.
it was almost impossible to do that in Canada.
Yeah, absolutely.
So. So yeah, I mean, I think that there are quite a lot of benefits. We know that there are a lot of benefits for children, and that's something that maybe we should in terms of the regulatory mechanism that we have, maybe we should think about it, and maybe we
Mhm.
should bring it back, and there's no
Yeah.
reason not to have it.
I view it's enough disruption in diabetes and now the new data in Alzheimer's. If I were to then go to the federal government in the US and go, oh, by the way, I got another vaccine and I think everybody should have that one, I'd probably be shot down right now. So,
Yeah,
I keep it to the niches of designated benefit.
yeah,
But if drugs get approved and people realize there's other things that help their kids not lose school time from all these infections, and maybe, yeah,
yeah.
it's more platform protection than just 3-month protection from a flu vaccine, then people will be on board. People
Yeah,
will
I
be
mean,
on
this is a
board.
long-term protection that you could have it at least up to, I would say, 10, age of 10, 12,
Yeah, yeah, yeah,
And then, and it's the safest vaccine. So, I mean, there is no downside to this
yeah.
vaccine. We clearly know, except this rare situation and cases
Rare
that
things.
could
Yeah.
have it, with any type of the treatment or vaccine in general, it
It's pretty
does.
remarkable. So
Yeah.
we have 250 kids enrolled, pediatric trials, okay,
Yeah.
in diabetic kids. And these kids come in with their parents and they're all very excited, et cetera. And then when I tell the kids on vaccination day, I go, you know what, tomorrow you have to go to school. You're not going to be sick. You're going to got to go to school.
They don't want it.
So disappointed. What do you mean a vaccine that doesn't make me sick, doesn't give me a sore arm? I can't do soccer. I can't do lacrosse. What do you mean? You have no side effects. But it's really true.
Yeah.
The next day, everybody feels just fine.
Yeah. No, absolutely. Absolutely.
But I'll tell you, the pediatric population in general is very disappointed there's no side effects because
Yeah.
for me to write the excuse that they can have the next day off school, they're never sick.
Yeah. No,
Yeah.
I mean, yeah, I mean, there is no side effect.
Mhm.
It's really remarkable.
Yeah, yeah. And you're putting in bacteria. There's
Yeah.
there's
Yeah.
no side effect.
No,
So
exactly. Exactly.
there's some humor in what we do, right? It
Yeah.
breaks all the rules. It breaks all the rules. So,
Yeah. But I think
okay, well,
it's all boiling down. I think if we have a better understanding of how mechanistically these things works, then I think we will have also better opportunity for introducing like a vaccine like BCG back into the community.
yeah.
Still, we are struggling to know how does it work. I mean, we have the pieces here and there, and like why BCG work against TB in neonates and
Mhm.
children, but not adults. I mean,
Yeah.
so now we are getting the pieces here, and we know like
But maybe
unconvinced.
they have to run the trials 5 to 8 years, and
Yeah.
nobody's willing to sit around or get a federal grant
Yeah.
to go
No.
5 to 8 years.
Exactly, exactly, and then we discover other immune cells, like like in this case for the neonate BCG vaccination, we think it's all about like the major protection coming from the gamma delta T cells, which
Oh,
are
wow. Very different.
unconventional.
Yeah.
Unconventional T cells is kind of like innate like T cells,
Hmm.
and
Hmm.
we haven't like really tried to understand the other possibilities for developing vaccine against TB. So
Hmm.
so there are lots we could learn if we could understand the mechanism
Either
and then try to.
BCG or DB. They're
Yeah,
both very
exactly,
synergistic.
exactly.
Yeah. No, I think it's fascinating. I think it kind of stems from an old-fashioned field, but the data is so much fun because of the
Yeah,
potential for benefit. So it's a interesting thing. The old is actually more interesting than we ever thought.
exactly,
Yeah.
exactly.
Well,
Fantastic.
I want to thank you for joining us
Yeah,
today in
thank
this
you very much.
podcast.
Yeah.
Yeah, and the
Th-
many contributions of your work on
Thank
TV
you.
and BCG. It's so important. And I also think that it's amazing in this field of how many of the answers are very similar. There's a lot of fields where the answers are all diverse and they don't overlap. But actually, in the TB and BCG field, there's so much synergy and so much overlap of these new lessons.
Absolutely.
So that's I think really novel too, if you've been in science for a while. So
Absolutely.
here's to our listeners. If they want more information on this podcast series or the BCG clinical trials, just go to www.faustmanlab.org and also look at Maz's website to learn more about what he's doing up in Canada. And I think we're all freezing today, whether you're in Boston or in Canada. So hopefully we get through this winter and move on to the warm summer climate. Thank you again.
Yeah, thank you very much for having me. It was absolutely fascinating conversation and great
And I
discussion.
learned too.
Me too.
It's all good. You
Yeah,
think
yeah.
scientists know everything? No, no, not at all.
Fantastic.
Okay,
Great.
thank
Thank
you.
you. Bye.